A very good morning and warm welcome 10th standard for your online teaching learning session. Yesterday in the live session I have discussed lesson number 6, animal classification. Now the part that it focused on was the criteria, based on which the new system of classification has been performed. And I also gave you the tips of how these criteria will help you to remember the characteristics of every phylum, and also you will have to remember one organism from each phylum, right? Such tips I have given you in the videos of the lesson as well in the yesterday's live session. Now since these criteria are very important and the base of your lesson, on the demand and request of many students, I am resending this part of the lesson once again. And now when you have to study this, I would request you all to keep your textbooks in front of you. Focus on what I am saying and the diagrams given in your textbook for the better understanding. Students, science is a subject where practical knowledge is very, very important. Now, since due to the situation around us, we are unable to perform practicals. But yes, these diagrams will help you a lot in understanding and learning and remembering the criteria, the characteristics of different phylums. Okay, so once again, let us understand this criteria and make our study easy and interesting. Whatever classification we studied was in practice till a new 5-kingdom classification system was introduced by Robert Whittaker, wherein all the multicellular animals are included in kingdom Animalia. This we have studied in lower grades. Okay. Now this system of classification is based upon some criteria like body organization. How is their body organized? Then body symmetry. How is their body divided? Body cavity, germinal layers. Is their body segmented or no? And many more such criteria. So based on this method of Robert Whittaker, we would be studying this new system of classification. This I explained you before, while studying the history of classification. So first criteria is grades of organization. Now we have studied that body of animal is made up of multi-million cells, right? Now here in a multicellular organisms, we all are aware and we have studied in 9th standard that different cells are responsible for performing different functions in our body. But whereas in unicellular animal, body is made up of single cell and all the functions are performed by that same particular cell. This you can directly relate to amoeba. Okay, the digestion, egestion, every process is done by that particular single cell. Now, body organization of this unicellular organisms is termed as protoplasmic grade organization, right? Before that, just look on your screen. You have been given how gradually the grade of organization is goes on increasing. First, protoplasmic grade, wherein unicellular animals have been grouped, where the one particular cell performs all the functions of the body. Then cellular grade organs, tissue grade organisms, tissue organ grade organisms, and organ system grade organisms. Now this is how your grades of organization have been studied and they have been put into this order. Now let us see, I told you about the protoplasmic grade organization. Amoeba, paramecium are some examples of that. Single cell performs all the functions of the body. Proceeding to the cellular grade organization, a higher than protoplasmic. Here, the tissues are not formed. Okay, So their body organization is cellular grade organization. There are multiple cells but tissues are not formed yet. So they are grouped under cellular grade organization, your phylum Porifera. And one of the organism from Porifera is Cycon. This will be studying further. Then we come to next grade higher, cell tissue grade organization. Cell tissue grade organization. Here the tissues are formed, the cells come together to form a group of tissues wherein they'll be performing the functions of the body. So those animals are grouped under cell tissue grade organization. Your example is Cnidaria phylum, and jellyfish is one of the organism. Now if you go back to the phylums of non-chordate, first you had Porifera, then you have Protozoa, then you have Cnidaria, right? So here when you are studying the body organization or the grades of organization, you will recall at the very first you had animals from the Protozoa. Then you had animals in the second grade, that is your cellular grade organization, you have animals from Porifera to study. In the third grade, in your cell tissue grade organization, you have a phylum higher, that is Cnidaria. So this is how evolution has taken place, okay, and the animals have become more and more developed as to the previous ones. Clear? Then you have tissue organ grade organization. Here the name itself suggests a group of tissues are formed. Tissues are organized to form some organs, but some complete organ system is not formed. Okay, so those are your tissue organ grade organization. Then here you can have flatworms. Now flatworms belong to your phylum Platyhelminthes. Platyhelminthes is on number 4, a higher phylum than Coelenterata, right? So this is how students classification is being studied. The last one is organ system grade organization. Here your organ systems are developed and they perform different functions of your body. Example, crab, frog and human, as you can see on your screen, right? So the very first criteria of animal classification, grades of organization, is very much clear. We shall move to next, that is body symmetry. Now, in any imaginary sense, if a body of any animal is cut through the imaginary axis of the body, I repeat, imaginary axis, sometimes it may, and in many cases it may not produce two equal half. Okay, now depending upon this property and this criteria, there are different types of animal body. We will look asymmetrical body, radial symmetry and bilateral symmetry. Now asymmetrical, clear that there is no symmetry, there is no exact half, there is no particular fixed bifurcations that you will be getting. So here there is no any such imaginary axis of the body wherein through which we can get two equal half. Very common example is your amoeba and paramecium and some of the sponges. Coming to radial symmetry, in this type there is an imaginary cut that passes through the central axis but through any plane of the body. You can see the picture of the organism on your screen. That will give you exact two half. Starfish is your good example. Now in this case, there are 5 different planes which will pass through the central axis of the body, through which you can get two equal half. Okay, that is your radial symmetry. You can see the 5 different plane. Okay. And the third one is bilateral symmetry. In this type of body, there is only one imaginary axis of the body through which you can get two equal exact half. And here the organisms are insects, fishes, frog, birds, human. Okay. Now that doesn't mean you will imagine an axis on your own body or someone else's and you will start cutting through it. Please don't do that, or keep drawing lines. Please don't do that. It is just for your understanding, and you can just imagine, or you can see videos or pictures on Google for better understanding. Okay. So I hope I am very much clear till now. It's very easy to understand. Just listen to me and go through the textbook and the points given in it. You will easily understand it. Now, when you study the multicellular organisms, there are these germ layers which are formed during the initial period of your embryonic development. We have studied how embryos are formed from the zygote, how zygote is formed from the fusion of male and female gametes, right? So when this embryonic development is taking place, where there is a group of cells, gradually these group of cells mature into these germ layers, and these germ layers then gradually develop into your tissues, then further development of your organs, and organ system takes place. Now there are two types of germ layers. Okay, on the basis of that, you have two different types of organisms, diploplastic and triploplastic. Diploplastic organisms will be those who have only two germ layers, that is endoderm and ectoderm. Endo is the outer one, endo is the inner one, and ecto is the outer one, right? Endo means inside, ecto means outside. Whereas the triploplastic organisms will be having three germ layers. You can easily understand by the word di and tri. Di means two in biology and tri means three. So we higher animals have our trypoblast. We have endoderm, the most internal germ layer, mesoderm, the one in the center, and ecto, the one at the outside. Okay, you can see the difference looking at the diagrammatic representation given on your screen. So when you study these animals, some animals will have only two germ layers. Okay, your cnidarians, the third phylum of non-chordates. Okay, otherwise in most of the remaining animals you will have these three germ layers, that is endoderm, ectoderm, and the center mesoderm. Clear? The fourth criteria is body cavity. Now, cavity is also called as coelom. What is the coelom? It is the space between the body and the internal organs. Okay. Now, again, this is formed, or this is present from the time of the initial period of the development of embryo. Okay, this body cavity is formed, you can see. Now, this is formed from the mesodermal layer. Clear? So those would be your high animals. The triploblast, yes or no? Now, whichever phylum comes after Annelida, that we will be studying in detail when we start with the non-chordates. So, all the animals that are present in phylum Annelida and all the phylums or all the organisms that are present in the phylums after the phylum Annelida would be having this body cavity. Right? So these animals which have true body cavity developed from the mesodermal layer, they are called as eucoelomates. Okay? Eucoelomates. Eu means developed. It comes from the word eukaryotic, if you all remember. Good. Now, sometimes body cavity is absent. So, this you will get to see in the animals from phyla Porifera, Cnidaria, and Platyhelminthes. Don't get confused with the animals. You will be studying which animal is present in which phylum. Right now you just understand body cavity is present in which phylum and it is absent in which phylum. Whereas in many of the cases, okay, such as Aschelminthes phylum, they will have the body cavity, but it is not formed from either of the ways that is given before. Just like eucoelomate or acoelomate, right? So these animals are called as pseudocoelomate. Okay, it is pseudocoelomate. So the ones with the true body cavity, those are eucoelomates. The one who do not show presence of body cavity, those are acoelomate. That is, coelom is absent, cavity is absent. And pseudocoelomate, it is not actually true, but it is not absent as well. Okay, I hope this characteristic is understood by you all. Let us see the next one. Okay, now before we move to the body segmentation characteristic, let's just look at the pictorial presentation. See, the digestive tract, which is formed from the endoderm. I said you that from these you will have your tissues, your organs and organ system being developed. Body is covered by ectoderm. Ecto means the outside layer. Okay, and from mesoderm you will have the tissue layer lining your cavity. You can see the cavity as well. Coming to pseudocoelomate, body is covered from the ectoderm. The muscle layer formed from your mesoderm. Okay, and the false cavity that you can see in the pseudocoelomate. And coming to the acoelomates, I hope it is very clear the cavity is not present. Okay, tissue filled region that is formed from the mesoderm. The last characteristic is your body segmentation. Now, when you choose this feature or this criteria to study the animal, you will just have to superficially look at the animal and you will be able to distinguish. You can see small segments or small units like throughout the body of the animal. Okay, so this what you can say, the body of the animal, when it is divided into your smaller units or smaller segments, okay, that body is called as segmented body. The organism on your screen is very commonly seen, and in this season you will get to see in huge numbers. Earthworm, right? You need not have a microscope or magnifying glass to look at this or to study this organism. You can just superficially look at this earthworm and you can identify the segments present on its body. So you can say earthworm has a segmented body. Then you will also get few animals from phylum Annelida which would be having such segmented body. So the ones those are having segmented body, these are placed in the phylum Annelida. Okay, so on the basis of these 5 different criteria, the animal classification is done, and then you have your total chart ready to study and understand. See, this way the organisms are studied with the help of the criteria that we studied, and then the animals are classified and placed in particular phylum. So all the animals with the cellular grade organization and asymmetrical body, okay, they'll be placed in the phylum Porifera. Then the animals which show radial symmetry and are from the tissue organ or organ grade system, they would be placed in the phylum Cnidaria, right? Then and also there will be some organisms which belong to the tissue organ system grade body organization, but they show bilateral symmetry. Then you have body cavity to study. Okay, so as the animals or the phylums get going higher and higher, you would be able to see more and more criterias being present. The animals would be much more developed. Okay, and then this is how the classification is done from lesser developed to the totally developed animals. So that is why I told you that you will have to remember the order of the phylum. You cannot mess up with that, because the lesser developed or the least developed organisms are placed in the lower phylum, whereas the totally or completely highly developed organisms are placed in the topmost phylum or the topmost class that we'd be studying now. Okay, so I hope till now whatever we have studied is very much clear to you all. And if you remember this criteria, this method of classification, I am sure this is really going to help you in studying the characteristics of the animals, because then it would be easy for you also to differentiate or to place the animals, okay, as a scientist have studied. I hope now your doubts are cleared and the criterias are easy for you to understand and remember. If still you all have any doubt, then you can ask in the next live session without any hesitation. Okay, but make your base very clear. Thank you, students. Stay safe, stay home, take care.